I have the pleasure this morning of introducing Dr. Harris. And I had to prepare something here because his CV and everything was so long. I was trying to summarize it here. So anyway, Dr. Harris received his BS from Gordon College in Massachusetts, followed by his MD PhD training at UMass Worcester. He is currently the chair of the Department of Dermatology, founding director of the Autoimmune Therapeutics Institute, and founding director of the Vitiligo Clinic and Research Center at UMass. He's been in Massachusetts for a long time. He has over 86 original publications in journals such as the Journal of Immunology, JID, Science Translational Medicine, Journal of Experimental Medicine, JAMA, and Lancet, among many, many others. He's been recognized for his basic science research by the Presidential Early Career Award for Scientists and Engineers, received the AAD Marion Sulzberger Award for conducting basic science research that had a significant impact on the clinical practice of medicine, and was elected to the American Society of Clinical Investigation in recognition for his contribution to clinical advancement through translational research. So please join me in welcoming Dr. Harris today for his talk on translational research in vitiligo. Thanks so much.
Thank you. I'm happy to be here. I'm hoping we can get the Zoom piece off. I'm not sure. Usually if you move the mouse away, it will go away. It's just then you won't see the top of my slide.
Do you want to move this out?
They have these on Facebook all the time. There are these things that pop up and things, what you didn't know about Zoom. I always read those because being able to navigate Zoom nowadays is really important. Thanks for having me. This is the first time anybody has ever said, don't worry if you run over a little bit. So I'm going to speak for 2 or 3 hours today. Okay. I could talk a long time. I promise I won't. But I would encourage you to please interrupt me with questions. If anything's unclear or if you just have a neat idea you want to talk about, it's my favorite part of presenting is really interacting with listeners. So today I'm going to talk about translational research in vitiligo and really focus on a lot of what we've done in the lab to understand vitiligo and develop new treatments for the disease. Translational research to me really is studying human tissues to understand mechanistic concepts, ultimately to find new treatments. Translational research has a lot of different definitions. And I believe that we really are launching a new era of targeted treatments. I think that that's the next phase where we can go beyond topical steroids and calcineurin inhibitors and UVB phototherapy to start to target pathways that drive this disease specifically using, hopefully someday, biologics. If anybody's going to hear the talk tonight, it'll actually be quite different. It'll be much more clinically focused, talking about recognizing unusual forms of vitiligo, using conventional therapies, and then moving into new therapies. And so I think it's worth hearing both if you have the time. Let's see. Now this isn't working. Try this. All right, there we go. So these are my disclosures. There's a lot of things up there. I work with a lot of pharmaceutical companies to understand vitiligo and help them develop treatments. I have no actual conflicts yet because there is only one FDA-approved treatment for vitiligo. Does anybody happen to know what it is? Almost nobody ever gets this. So I'd be impressed if you can tell me the one FDA-approved treatment for vitiligo. No? It's monobenzyl ether of hydroquinone. So that is the one drug that the FDA saw fit to approve. Michael Jackson used it to depigment his skin. The problem is the only FDA-approved drug makes disease worse. So we have to do a lot better than that. And therefore, I'll be discussing off-label drug uses because there's nothing out there. We do anticipate one drug will be approved this summer, and it'll be the first one. So vitiligo is really common, about 1 in 100 people. That's a lot of people. And 50% of patients develop vitiligo before the age of 20, 80% before the age of 30. So a lot of people are getting it as kids, and it doesn't spontaneously resolve. So patients have this for the rest of their lives. As a clinician, that means we're managing these patients over decades of their lives, optimizing their management. But as a scientist, it gives us an opportunity to study this over that same period and look at waxing and waning and try to understand what causes the disease. It also has a lot of disease associations like type 1 diabetes and others. This is a patient that I like to show who has vitiligo on her hand here and on her abdomen. She also has a insulin pump for her type 1 diabetes and an ostomy bag for her Crohn's disease. So we think as we study vitiligo and we find pathways that drive the disease, we're really studying all autoimmunity. And this is an opportunity to develop treatments for diseases that are much harder to study as well. So this is how it starts. It's little macules of depigmentation here and here. Those grow and coalesce into larger patches. And this is a typical presentation on the hands. It can become quite widespread. It can through the universalis type, it can depigment the entire body. This can really be psychologically devastating for patients. And the impact on the quality of life is the same or greater than those with psoriasis and atopic dermatitis, believe it or not, for different reasons. Typically not symptomatic, but very visible. The great news about vitiligo is that it's reversible. So this used to be a white patch. Now you see little brown spots within that white patch. This is how it becomes reversed. And those little brown spots come from the hair follicles because the melanocyte stem cells live in the bulge of the hair follicle. And this has really important implications for treatment. So hair-bearing areas of the skin are very responsive to therapy. Glabrous skin, completely unresponsive. And that's important. So this is actually a typical response to UVB phototherapy over the course of a little over a year. And at 6 months, I was looking at her neck and chest and said, wow, you're responding fantastic. This is a great response. Great job. She said, what about my face? And I said, what about your face? And she said, I used to have vitiligo all over it. So by 6 months, she had gotten so much of her pigment back on her face, I forgot she had vitiligo there. And this is not an unusual treatment response. So this is important to communicate because usually I'm the 5th or 6th dermatologist that my patients have seen. And they said that the other 5 told her that there was nothing that they could do. So we're trying to stop that. All right, so more tonight about different presentations, conventional therapies, and where we're going. But now I want to talk to you about how we've really worked hard to try to understand this disease mechanistically to develop new treatments. So this patient came into my clinic having received a kidney transplant for unrelated reasons. He was on systemic tacrolimus, and he repigmented faster than anybody I'd ever seen with vitiligo. Really telling us that if we were able to systemically immunosuppress all of our patients to kidney transplant levels, their vitiligo would do well. But we know that we can't do that because of the side effects of this treatment. But we thought, well, could we develop targeted therapy that's both safe and efficacious? We can do this in psoriasis. We can target a single cytokine and reverse psoriasis long-term in almost everybody. So the hope was that we could be able to do this for vitiligo. So the approach that we've taken in the lab is to integrate clinical research, clinical trials with basic science in a mouse model, as well as translational research using human tissues to really understand mechanisms so that we can develop better treatments. So I'll tell you a few stories today. The first story is a bench to bedside story starting about 10 years ago. I developed a mouse model of vitiligo. It was really an adaptation of a melanoma immunotherapy model. But this is briefly what we do. We take a mouse that has black skin and black hair. It's essentially on the B6 strain, but has a mutation that maintains melanocytes in the epidermis, interfollicular epidermis as well. We adaptively transfer in CD8 T cells from a TCR transgenic mouse that recognizes GP100, which is a melanocyte-specific antigen. And then once those cells are inside the mouse and engrafted, we activate those cells either with a dendritic cell vaccine loaded with GP100 to activate them, or a vaccinia virus that expresses GP100. Either way, it activates those T cells and gets them angry, and then they run around the mouse looking for melanocytes. And essentially, we end up with a mouse that has white spots on the tail, the feet, the nose, and the ears, and we can quantify that into a total severity of vitiligo. So we began by just looking at gene expression in the skin of those mice. So here the white bars are mouse vitiligo skin, the black bars are human vitiligo skin. We're just looking at gene expression. Clearly, we see a loss of melanocyte transcripts that you'd expect when melanocytes are dying. But then we see a very strong interferon gamma-driven signature. And that's important because we see no type 17 inflammation that you see in psoriasis, no type 2 inflammation that you see in atopic dermatitis. And that's, of course, really important because we've got great drugs to target those other 2 pathways. And that's why those drugs don't work in vitiligo. There's nothing happening there. And in fact, both categories of drugs not only don't work for vitiligo, but they induce vitiligo and they exacerbate vitiligo. So we've seen now 7 patients actually treated with dupilumab who've either had vitiligo onset or exacerbation shortly after the drug. That resolved when the drug was stopped. So pretty important, and highlighting the fact that we've had nothing to treat vitiligo patients. So we needed to look into the interferon gamma signaling pathway. We spent a long time doing that. It's always sad to show one slide that represents 10 to 15 years worth of work. But the interferon gamma signals through its receptor, through JAKs1 and 2, STAT1, CXCL9 and 10, gets secreted that recruits more T cells through CXCR3. And we've targeted every member of this pathway genetically through knockout mice that prevents vitiligo, or using antibodies that target the soluble mediators that both prevent and reverse vitiligo, and as well as JAK inhibitors that work for mouse vitiligo. But maybe more importantly than JAKs working for mouse vitiligo is that they work for human vitiligo. So this is a paper that we published in collaboration with Angela Cristiano and her group, where a patient was given oral ruxolitinib, and his vitiligo was reversed in pretty rapidly, in about 5 months. So this is before and after oral ruxolitinib. And this was done with Mehdi Rashigi, one of my fellows at the time, now has his own lab at UMass, doing translational research as well. And in addition, we had saved up this patient's serum for over a year. So he was actually enrolled in an investigator-initiated clinical trial to test simvastatin as a treatment for vitiligo. That didn't work, but he was put on placebo at the time, randomized, and we had collected his blood over that entire time and froze it. So once he started the ruxolitinib, we kept collecting his blood, and you can see that he has this very high level of CXCL10. This is the gamma-induced chemokine that we've been studying. And that was stable for over a year. But as soon as he started taking ruxolitinib, we saw that level drop, really telling us that not only is our oral JAK inhibitors effective in treating vitiligo, but they seem to be working the way we'd hypothesized by blocking interferon gamma signaling, reducing downstream effects, and allowing repigmentation. So based on this and another case report by Brett King, testing Xeljanz as a treatment for vitiligo, and some more by David Rosmarin at Tufts using topical ruxolitinib, all showing efficacy, really launched this new era of testing JAK inhibitors for vitiligo. These are ongoing or recently completed clinical trials. And this is Zeni Abbas, who was a fellow at the time that helped run these trials in the lab. Now she's a second-year dermatology resident with us. So first I'll tell you about the INSIGHT clinical trial. This is a phase 2 clinical trial testing topical ruxolitinib as a treatment for vitiligo. Patients were randomized to a bunch of different topical doses as well as vehicle control for 6 months. After 6 months, the vehicle controls were re-randomized to a higher dose of drug. And then after a year, everybody was put on the highest dose of drug and given the option to use UVB phototherapy. And these are the results. So the first half was published in 2020 in The Lancet. This is just the first year of data. And now we've been following them. The trial was extended to 2 years. So we have 2 years of data. You can see, first of all, the primary outcome was the F-VASI 75. That's 75% improvement in the vitiligo on the face. And there was a 0% placebo rate. So rarely do you see that in a clinical trial with 0 placebo. So all that you're seeing here is pretty significant. You can see the green bar, the F-VASI 75 after 6 months was achieved by 30% of patients, after a year, over 50% of patients, and then after 2 years, almost 75% of patients. So a few things here. The JAK inhibitors work. Topical ruxolitinib works for vitiligo. It is slow, just like everything. So people who are used to running psoriasis clinical trials, atopic dermatitis clinical trials, patient comes in literally the next day on dupilumab and tells you that they're not itchy anymore. That's not what's going on in vitiligo. So we're looking at 6, 12, 24 months for full treatment. But patients are getting better, and this is really exciting for them. And the patients are staying in the trial because they're getting better. So it's worth the wait. It just takes time. So what we're showing, the primary outcome is facial vitiligo, because there's a high density of hair follicles in the face, much faster to repigment. but it also works in the body. So you can see after 2 years, over 60% of patients getting 50% improvement on the body, and almost no improvement in glabrous skin as we'd expect. Then this provided the rationale for a phase 3 clinical trial in over 670 patients all over the world, now enrolling down to 12 years old, and allowing us to see adolescence effects and just testing the highest dose, 1.5% BID. And fascinatingly, as a mouse biologist and understanding the variability in experiment to experiment, this was almost exactly the same results. So phase 2, 30% achieved the primary outcome. Phase 3, 2 different parallel clinical trials in 300 patients each, also 30% achieved the primary outcome. So this is really robust data. The one difference is that there's a higher placebo rate now. We think that that's because they needed to use lots more centers who didn't really have expertise in vitiligo, and so the outcome was a little bit more messy in the placebo. But again, 30% achieving an F-VASI 75, really impressive. Data works. Yep.
You make a good point about the necessity to be patient and to go for a long time in therapy and watch. So, do you have any sense of what the response is like if people have been like on current therapies?
Yeah.
Topical calcineurin inhibitors, UVB,
Right. Yep. So questions about the comparing conventional therapies, the current therapies that we have, and how long that takes as well. And it's also very long term. So a lot of the times, the reason why patients come to me is because they said, "Oh, I went to a dermatologist. I used this treatment for 3 weeks and didn't see anything." And I said, "Wow, that you should use it for 3 months before you'll start to see something." Even those tiny perifollicular brown dots. We say that can take, and actually, I don't know if I have it here, my previous slides. Now, I'm not showing the earlier data. If you read this paper, some patients started responding by 8 weeks on topical rux, but some took much longer. And so I think that's and even on phototherapy, I say, don't even look for any improvement for 3 months. Use it, and we won't know for 3 months. Some of the patients in here, the quote non-responders, were actually slow responders, and it took them, they had a lag of about 6 months. So definitely all the treatments take that much time. And if you think about it, for psoriasis to get better, all you have to do is remove the inflammation, and the epidermis continues to turn over in a couple of weeks, and then you're calm. Here, we've got to get melanocytes stimulated to proliferate, migrate, differentiate, start making pigment, and transfer it to the keratinocytes. And I think that that's why it takes so long. So if there's, I really encourage people to become vitiligo experts, because this is, 30 years ago, psoriasis was so exciting. We've got all these psoriasis experts now because it's so much fun over the past 30 years. The same thing's about to happen for vitiligo, but you've got to be a patient person and make sure that you prepare your patients for that too.
Well, I guess my point is, the JAK inhibitor is not a really target. It's probably less safe to tackle.
True.
We have tackled.
Yep. Yeah. So are JAK inhibitors really in advance, right? That's a big question. So a topical JAK inhibitor, it does seem to work faster than topical tacrolimus. They haven't been tested head to head. We won't know for sure. It's just that I've been super impressed when I see people respond. They're just responding faster. JAK inhibitors aren't working on areas that the others don't work on. That's also another question, right? Glabrous skin still not responding to this. And do we need another topical when we've got topicals that already work? It's a great question. I'm much more excited about getting a systemic JAK inhibitor because that we don't have. We don't have a systemic treatment. Right now, the only thing we have is narrowband. So I think that would be a real advance. Proof of concept, though, this is pretty good to get started. Yeah. So it is in fact not a dumb question. The question is about laser hair removal and what the impact would have on vitiligo. We were just talking about this in my clinic this week on Tuesday, and noting the fact that there is absolutely no data, and we get asked this question all the time. People are like, "Can I do laser hair removal?" Theoretically, if you remove the hair, you remove the bulge, and you remove the melanocyte stem cells, then you're not going to be able to repigment. We just don't have that data, and we don't know. Permanent hair removal is rarely permanent, as you probably know. So the stem cells maybe aren't removed when you do that. So we don't know. The other question that comes up is if you if you do laser hair removal, does that Kebnerize vitiligo? Because you're blasting, this pigment-rich thing. Does that attract inflammation? We don't know the answer to that either. So it's a question that comes up a lot. One time I just went onto the Facebook vitiligo group and said, hey, has anybody had laser hair removal and what did it do to your vitiligo? That was like, I think the best way to answer this question. And got lots of responses, lots of people who did it and didn't have any problems. But your specific question about responsiveness to therapy in the distant future is really hard to study, and we don't know. Yeah. All right. So the next study was indeed an oral JAK inhibitor. This was done by Pfizer, testing ritlecitinib in a phase 2b clinical trial. This is a JAK3 TEC inhibitor, and patients were randomized to a bunch of different doses orally. The difference with this trial and the INSIGHT clinical trial, it's important to remember, is that they required disease activity. So you had to have active disease as vitiligo patient to enroll in this trial, which I thought was a bad idea, and I advised them against, because active disease is actually really like a freight train running, and it's difficult to stop. So it's a much higher bar. But they in their minds, they thought, well, it should be active, and then the immune system's working because this is inhibiting the immune system. When in fact, stable vitiligo also has immune function acting as well. We'll show that. So in this study, we saw a dose improvement that correlates with dose. We could see that the highest dose responded the fastest. Everybody at 6 months was re-randomized to the highest dose, and they continued to get better. But after 6 months, those achieving the primary outcome, same as in the previous study, was only 12% instead of 30%. And so they actually were very disappointed in this and put this on the shelf, and they're not doing a phase 3 clinical trial right now. It's very possible. So it's possible that this drug isn't as good as topical Rux, right? But it's also possible that, based on the fact that they only enrolled active patients, that 12% is pretty impressive. That if you only take the hardest to treat patients, achieving the primary outcome in 12% was good. And based on that, we reanalyzed the data. This was just presented at the AAD a couple weeks ago. If you split the active lesions and the stable lesions in the patients enrolled in this trial, you could actually see that those who had an active lesion on placebo got worse. They lost pigment. And those who had drug with active lesions, the lesions stabilized. And the stable lesions in those patients, the placebo didn't change at all. There was no improvement, no worsening. But those who were on drug actually repigmented. So this really outlines the importance of enrolling for your clinical trial, thinking of inclusion criteria, and keeping this concept of active vitiligo versus stable. The drug actually worked really well because it stabilized active lesions and prevented them from getting worse. And then the stable lesions got better. So if you split this out, I kind of think that the Pfizer trial would have had the same result as the INSIGHT trial and not been worse.
Is there any evidence for a difference in immunological response in the active phase?
So I don't think so, and I'll show a little bit of data about how we've started to dissect that. I'll show that a lot. Let me know if I don't answer your question in a couple slides. So that's the bench to bedside story. We found interferon gamma expression, interferon gamma signaling, downstream pathways. You can inhibit that using JAK inhibitors. They've been shown to work in clinical trials. But can we do better than that? So here's a bedside to bench and back story that started with the observation that vitiligo patients relapse when you stop treatment. Thierry Passeron published this great paper in 2015 reporting that about 40% of vitiligo patients relapse when their treatment is stopped, regardless of what treatment they've had. And incidentally, you can use topical tacrolimus just twice weekly, and you can reduce that relapse rate to under 10%. So that's a bona fide maintenance therapy if you want to use a maintenance therapy in your patients. But I see this all the time, and important to me as a physician is not only does it relapse, and I've got to think about that and how to optimize their therapy, but it comes back in the exact same place it was before. So it relapses in the exact same locations, previous lesions. And this is a patient I treated who got pregnant, and she responded beautifully to UVB, then got pregnant, stopped UVB because she got busy, and a couple of years later came back to me with exacerbation relapse on her abdomen, the same place. So we went back to the bench and said, we want to know if we can figure out why disease relapse. What's the source of this autoimmune memory forming in the skin? How does the skin know it's supposed to have vitiligo after it's improved? And we weren't the only ones to go and ask this question. So 3 other labs from around the world also asked the same question. And the 4 of us all came to the same conclusion, which tells you that the data is probably pretty robust. And so I'll outline that here. So I already talked about the fact CD8 T cells infiltrate the skin. They find melanocytes. They make interferon gamma. That interferon gamma induces CXCL10 secretion from the keratinocytes and brings more cells in through the receptor CXCR3. And this creates a positive feedback loop that drives the progression of disease. But it turns out that a subset of these cells also form what's called resident memory T cells. So these T cells upregulate adhesion molecules. They downregulate leave the skin molecules that cause them to exit. And they essentially set up residence in the epidermis forever. Now, this is supposed to happen during viral infection. This is where they're first described, where these cells go in, they kill the virus, they wipe it out, and they leave some cells behind just in case that virus ever comes back. But we all found that the same process is happening in vitiligo. These are autoreactive CD8 T cells. They form into resident memory. And when a melanocyte tries to grow back and they engage that melanocyte, they make interferon gamma, which induces CXCL10, and they recruit more effector memory cells from the circulation to kill those cells. So this is the point that we were talking about before. This is the progression of vitiligo. And this is the maintenance of vitiligo. So even stable lesions, if they're not getting better and they're not getting worse, the reason they're not getting better is because there's continued inflammation in there through resident memory. This is a much more subtle inflammation here. And so the progression of vitiligo when it's active is really lots of T cells making lots of gamma with lots of recruitment and growing that lesion. But when they stabilize, they become just this same process, same pathways, but just a subtle kind of low-level subclinical inflammation. And that's why both of these processes can be inhibited by JAK inhibitors. So stable vitiligo or active vitiligo. The conventional treatments can turn these guys off and allow repigmentation. JAK inhibitors do the same. But the problem is, as soon as you stop these treatments, those cells are still there. They wake back up and reinitiate the process and relapse. And we can see that in our mouse model. So if we take mice, treat them with JAK inhibitors, they get better. But if we follow the resident memory T cells, we can see that the number is not changed. So it's their function that's modified by JAK inhibitors, but not their presence. So we asked, could we actually target the resident memory T cells, remove them from the skin, erase the memory, and not only have a good treatment for vitiligo, but actually have a long-term, long-lasting treatment. So you can stop the treatment and maintain disease clearance. And long story short, Jillian in the lab, one of my fellows who has her own lab now at UMass, found that if we block IL-15 signaling, then we can not only repigment vitiligo, this is better than we'd seen with any of the JAK inhibitors that we'd used, but we can eliminate the resident memory T cells from the skin. So you block IL-15, the resident memory T cells go away. We don't know if they die or if they just leave the niche, but IL-15 is constitutively expressed in the epidermis, and that's kind of what tells the T cell, hey, you're in the epidermis now, upregulated adhesion molecules. So you block that and they disappear. We did lots of other experiments showing that if we just treat and block IL-15 for a short period of time, we can get repigmentation over months, showing that it's a durable effect. So you can block IL-15 signaling either with an antibody against the cytokine IL-15 or an antibody against the cytokine receptor, IL-15 receptor beta chain. And I applied to the NIH, the Immune Tolerance Network, to fund a clinical trial to test an IL-15 inhibitor for vitiligo. That's ongoing, but I had to recuse myself from that trial. I am not the PI of that trial because I was able to get venture capital funding to start my own company called Volaris Therapeutics, where we're targeting the receptor beta chain. And there's a bunch of reasons why we think targeting the receptor is better than the cytokine. But the Amgen drug is being tested as we speak, enrolling for a clinical trial, and Volaris will have our antibody in humans next year. So we're moving pretty quickly. So lots of exciting opportunities, we think, to develop actually a biologic to treat vitiligo. Yep. Yeah, it's kind of like, the question is if you treat for just short term and then remove the treatment, I kind of think of it like shaking up an Etch-a-Sketch. You can redraw a picture, it just takes a lot longer. So, we don't think that we're curing the disease, we're just erasing the memory from the skin so relapse isn't as rapid. But you can still get T cells from the lymph node rediscovering the skin and initiating a new lesion de novo, but that takes years. So the idea is we could treat for a window of 4 to 8 weeks every year and keep it completely at bay. That's the hope. Yeah. All right, so here is just a quick summary of this part. Resident memory T cells form. We can block their function by using JAK inhibitors or cytokine inhibitors, or we can block IL-15 and actually remove the cells and erase the memory for long-term treatment. At least that's what we see in the mice. So then we asked, well, can we do better than that? So I mean, that's part of academic medicine is always looking for better and learning more. So we wanted to learn more about vitiligo pathogenesis in higher resolution. And to do that, we kind of dug out an old technique that was developed at the NIH maybe 20, 30 years ago of suction blistering human skin in order to sample it. So we use a negative pressure machine that creates a suction on the skin, LED lights to heat up the skin a bit. You get a blister in about 30 minutes. Without the heat, it can take about 3 hours and hurts like crazy. So this is painless, no scars, no stitches. We don't need anesthesia. So patients tolerate it really well. This is my foot, so I know what it feels like. You can create these little blisters and we can go in and suck out the fluid and get all the skin cells to analyze. And I'm showing Jim Strassner, an MD PhD student in the lab, about to start residency this summer at Hershey, and Maggie Ahmed and Van, who helped kind of develop this technique in the lab so we could use it to study disease. So this is one of my patients reading a magazine, very comfortable with the suction cups on her foot and on her leg. And we can position these cups, the blisters themselves, right over little tiny spots. We call this confetti vitiligo, where there's 1 or 2 millimeter deep macules. We know that the T cells are active there. That's an actively spreading lesion. So we can sample those areas, and then we can also sample non-lesional skin as a comparison. And so this is showing that. We can also look at healthy skin, healthy, lesional, non-lesional. We create the blisters. We take the cells out. We can look at them by flow cytometry or single cell RNA sequencing, which I'll tell you about. We can take the fluid out. We can look at that by ELISA or proteomics or metabolomics. There's a lot that you can do here. It's been a great technique for us. We've blistered hundreds of patients now, and we've really learned so much from them. The other thing too is you can do a punch biopsy, 4 millimeter punch for analysis. But we can do 10 1 centimeter blisters. So it gets us 10 to 20 times more cells to study, again, in a painless, non-scarring way. So essentially taking the mixed fruit bowl and ordering it into fruit rather than blending it up into a smoothie like we used to do to look at gene expression in tissue, we now can look at each individual cell that we pull out of the skin. What we're showing here is gene expression in the cells on the right. And based on the fact whether it expresses keratin 5 or keratin 2 or loricrin, we can actually position the keratinocytes. So this is a keratinocyte cluster. We know these are basal keratinocytes and these are differentiated granular keratinocytes. We can find lymphocytes, melanocytes, dendritic cells, macrophages. And then we can actually take those lymphocytes and pull them out and subset those lymphocytes and find innate lymphocytes or adaptive lymphocytes where we're looking at CD4s, CD8s, T-regs, gamma deltas, and NKs. And so we can, number one, find these cells and characterize them. And then we can actually look to see what they're expressing and what they're doing differently in vitiligo. So this is a crazy big heat map that I'm showing you of ligands and receptors. So these are all the ligands and receptors in the human genome that change in vitiligo. And what the first thing that, and each column is a different cell type. So these are melanocytes, these are NK cells, these are all the keratinocyte subclusters, CD8 T cells, T-regs. And the first thing that really struck us is most ligands are made by only one cell type. So if you look at this whole series of ligands that are changing here, these are only made by melanocytes. And these are going to their cognate receptors, which are only expressed by one cell type as well. Right? So if you take a cell type making a ligand and find the receptor for that ligand on a different cell type, you know that there's a one-way conversation happening there. Cell A is talking to cell B through this receptor ligand pair. And what might be a little difficult to see is in each column, there's actually 3 columns, and that's showing healthy, non-lesional skin, lesional skin. So we can see how things are, each one is changing. So it's too hard for you to digest, but individually, but just look at the whole pattern and try to understand how vitiligo is causing all of these changes in the skin. So I told you about 2 pathways, interferon gamma signaling and then IL-15, right? And then here's hundreds more to start pursuing. It's a little intimidating. And by the way, this is Manuel Garber, our computational biology colleague at UMass. Those guys are worth their weight in gold. And Jake Galatly, his med student who worked, or his grad student who worked with my MD PhD student on this. So then they can take all these one-way conversations and create a network of conversations that define vitiligo. So each dot here in the network is connected to another dot that's a cell population speaking to another cell population. And the line in between them is the receptor ligand pair that describes it. And if the line is red, that means it's a conversation that's increasing in vitiligo lesions. And if it's blue, it's decreasing. So you can see some big categories. Class 1 expression, MHC1 goes up. It's not surprising. We know that happens when interferon gamma is around. That's how the T cells talk to their targets. So this is a very sensitive assay for looking at inflammation. But then we see a lot of chemotaxis, a lot of chemokines that are going up and changing. Chemokines attract cells and move them around the body, either into tissue or within the tissue. We see a few things going down, like Notch signaling. We talked about Notch just now. TGF beta and a number of other things that are really interesting. But for the paper, we focused in on chemotaxis because it's what we're good at. We know how to study it. We already talked about interferon gamma and CXCL9 and 10. We could take those chemokines and their receptors and blow them up into another heat map. Similar heat map as before, but much more simplified. And you can look down, maybe some of you have favorite ligands, chemokines, but I'll just highlight CXCL9 and 10 and their receptor CXCR3. We've been studying that for 10 to 12 years, and we saw that those come up as well. So nice confirmation that we see this pathway over and over and over again. And the chemokines are being expressed by keratinocytes, as we'd previously learned, here and here, but even more so by macrophages and dendritic cells. And so this is just another piece that we still have yet to understand what's their role in driving this disease. But then this really caught our attention. CCL5 and CCR5. These are thought to be a type 1 chemokine, so expressed on CD8 T cells that also participate and drive inflammation. But one thing that was curious is CCL5 doesn't change. So this is healthy, non-lesional, lesional. CCL5 isn't changing expressions, primarily expressed on CD8s. But CCR5 is being expressed by T regulatory cells, and it's going up in lesions. And this was a little bit curious as to why T-regs are making CCR5. Are they now becoming not T-regs because it's an inflammatory chemokine? Or are we wrong that it's an inflammatory chemokine and actually T-regs are using it to suppress? Either one of those could have been true. So here's just a blow up of that heat map now where you can see CCR5 going up, particularly on T-regs, but also on CD8 T cells. If we look by protein, so that's all transcriptional RNA stuff. If we pull the cells out of the suction blisters and we look by flow cytometry, we can see that most T-regs are making CCR5. And actually in lesions, they're making a lot more than in non-lesional skin. So the protein is showing the same thing that we saw with a transcript. So this is Kingsley, who was a grad student at the time. He went into the mouse and found that the T-regs are doing the same thing in mice. So in vitiligo, skin draining lymph nodes or ear skin, we can see that if they have vitiligo, their CCR5 expression on T-regs increases. And so this gives us an opportunity now to ask mechanistic questions. Why are they doing this? Is it because they're not good T-regs anymore, or are they actually using it to suppress? So Kingsley also did some neat studies where we labeled the CD8 T cells red that were going into the host mouse, and the host mouse made T-regs that were green. So we could look at interactions between CD8 T cells that cause vitiligo and the T-regs that are trying to suppress them, and they're green. So you can see in this fluorescent picture, green and red T cells looking like a Christmas tree, all clustering in the same areas within the skin. This is in the ear of a mouse. And it looks like the T-regs in the CD8 T cells are going to the same areas, like the T-regs are almost chasing them to shut them off. And when you look even closer using confocal microscopy, you can see that those cells are shoulder to shoulder and actually even reaching out and shaking hands. So it looks like the CD8 T cells are reaching out and touching the T regulatory cells. really suggesting that cell contact is how the Tregs are working here. And that in order for the Tregs to work, they've got to get right next to a CD8 T cell. They have to be able to find them. So that started to cause us to ask questions about how Tregs get around and how do they find their targets. Well, we also see this in humans. So this is immunofluorescence in human skin. Looks almost identical to what we were seeing in the mouse. The green Tregs and the red CD8 T cells directly interacting in that handshaking here. And by the way, the Tregs express CCR5, just to further confirm what we had found previously. So then Kingsley developed this mouse model of vitiligo that's similar to our other model, except that it's in a RAG knockout. So the RAG knockout mice don't have any of their own T cells. So we essentially either just put in CD8 T cells with no Tregs, and they get a lot of vitiligo. We put in CD8 T cells with wild type Tregs, and they suppress vitiligo pretty well. And if we give them CCR5 knockout Tregs, the Tregs don't work as well. So they get worse disease. And somehow, so now the Tregs seem to be using CCR5 for their suppressive function. Whereas again, we had thought that this was an inflammatory chemokine. So then the next obvious kind of thought was, well, CCR5 must get Tregs into the skin. That must be how they know how to find lesions and get in there. But what we found that surprised us a lot is the CCR5 knockout Tregs have no problem getting into the skin. They get into the skin just as well as wild type Tregs. So the migration piece of this was confusing. So next we used, this time, green wild type Tregs and red knockout Tregs together and put them in the host to see where they were going. Were they going different places? Were they behaving the same? And what we found were, again, the Tregs cluster like this. But in the clusters, we could see a lot more green cells than red cells. When we looked closer, we could see, again, a lot more green cells than red cells. So the CCR5 deficient Tregs got into the skin fine, but they couldn't find these clusters. They couldn't find their targets for some reason. So we think, still haven't definitively shown this yet, but we think that Tregs, once they're in the skin, they use CCR5 in order to find CD8 T cells, their targets, and be able to make contact with them and regulate them. Remember, the CD8 T cells in our heat map were the primary producers of CCL5. So this would be a great way for them to hunt them down and find them. But we still didn't know how Tregs got into the skin in the first place. And we wanted to. So you can see in our single cell data in humans, we can see Tregs are at much higher numbers in lesional skin than non-lesional skin. So they get there somehow. Something's telling them to go in there. We know that in the mouse, this happens as well. So a lot more Tregs go into lesional skin. Clearly, they're not enough to control it. You're still getting lesions, right? But we have other data that if you deplete Tregs, the whole mouse turns white. So the reason why we have spots in vitiligo is because the Tregs are keeping them contained. And so you need Treg infiltration to slow down the disease. But what's the cause? What's getting the Tregs into the skin? We know it's not CCR5. Well, we had previously published, as I mentioned, that interferon gamma drives CXCL10 that recruits effector T cells, the CD8 T cells into the lesion. And so if your CD8 T cells can't make CXCR3, you don't get disease, and they actually can't find their way into the skin. So the CD8 T cells don't find their way into the skin if they don't have CXCR3. And we see that Tregs follow the CD8 T cells into the skin. They actually, as soon as the CD8 T cells infiltrate, the Tregs infiltrate right after. So the obvious choice for us was, well, Tregs must be using the same path. They must be using CXCR3 as well. They're both using the same receptor. But that turns out not to be true. So if we take Tregs that don't make CXCR3, they work just fine. They still are just as immunosuppressive in our model as wild type. So CXCR3 was the wrong answer. But a friend of mine, Mike Rosenblum, and his postdoc at the time, Tiffany Scharschmidt, who now has her own lab at UCSF, discovered that commensal bacteria induce CCL20 within the hair follicle that promotes Treg infiltration during neonatal development through CCR6. So we're like, well, Tregs can use CCR6 to find their way into the skin during neonatal development. Maybe they use that during disease as well. So this was just an educated guess. And Kingsley also found that now if Tregs don't have CCR6, they don't work in vitiligo. So CCR6 knockout Tregs can't suppress like wild type Tregs. And when you look over here, it's because they can't get into the skin now. So this is completely different from CCR5. So this told us that Tregs use CCR6 to get into the skin and CCR5 to get to the location where they need to in order to suppress. All right, so here's another summary slide. CD8 T cells, we talked about interferon gamma, CXCL10. We can use JAK inhibitors to inhibit them. They convert into resident memory T cells from IL-15 signaling and maintain the disease that way. And Kingsley found that CD8 T cells now use CXCL5 to find their targets and CCR6 to get into the skin. So trying to build, if I showed you this as my first slide, everybody would have just glazed over, but hopefully now in pieces, you're able to kind of understand how this all works. The other advantage now with blisters, we talked about single cell RNA sequencing. We can use proteomics and metabolomics on the fluid. This is not very advanced, but here is a volcano plot looking at proteins using proteomics in the fluid. And again, just to verify, this is CXCL9 and 10 again. So we just see that pathway keep coming up over and over and over again. Now we're seeing the protein in the human blister fluid, giving us lots of confidence in the pathway. And then lots of other things that we can study as well. We're still trying to dissect and understand what their role is. But the great thing is being able to use proteomics and crossing that with our single cell RNA sequencing to really understand and learn more. And then we have similar volcano plots with metabolomics. So T cells have to create energy and they burn metabolites to do that. So we see some metabolites going up and some metabolites going down. And our goal is to integrate all of this into a real good understanding of what drives vitiligo. All right, and in the last 10 minutes or so, I just want to tell you about some opportunities to develop novel treatments for vitiligo beyond JAK inhibitors, beyond IL-15, potential durable treatments, biologics, to some new ideas on how to do that. I call this right place at the right time because it was really being in the right location to learn about important science that influenced this, but then also delivering the drugs to the right place at the right time. So the first thing we'll start out with the Nobel Prize for RNAi by Craig Mello at UMass in 2006. And at the time, everybody thought this was going to be the way we treated every disease from now on. RNAi allows you to target anything. But the problem was that RNA is not stable in vivo, number one. So RNases chew it up. It doesn't last very long at all. The other thing is getting it to the right place. So getting it to the right organ, the right tissue, the right cell type, getting it into the cell is really critical. And so for 14, 15 years, we really haven't been able to turn this over into a therapeutic just because of its instability, volatility, and then inability to target it. But Anastasia Kvororova, who is a faculty member here at UMass, is a chemist. And she was the chief scientific officer for RXI, which is a company making RNAi for therapeutics. And in 2019, she published that she was able to make two modifications to RNAi. One, to make it resistant to RNase, and then another to allow its delivery to various organs and tissues in vivo. It was selected as Nature Biotech's 25 landmark papers in 25 years, so pretty important. And this really supported Atalanta to get a $110 million Series A to target neurodegenerative diseases. So this is essentially what she did, and she's up there, and Tang, our shared postdoc, is here because he's interested in using this to target skin now. But essentially, the RNAi backbone was completely modified so that it's not RNA anymore, and it can't be chewed up by RNases, completely resistant to RNase, but still engages the RISC complex to knock down its target. So that's the first piece. And the second piece is the dianophore there that you can see in blue. This is a hydrophobic molecule that will be put on and conjugated to that RNAi that does two things. Number one, it self-delivers across the cell membrane. So when a cell sees this, that hydrophobic molecule pulls it into the cell. And number two, it also helps its distribution in vivo to get to different tissues, depending on what the dianophore happens to be. So the dianophore could be vitamin D. It could be cholesterol and any number of other hydrophobic molecules. And there's really no way to know which one's going to do the best for, which one's going to get it into the skin or the brain or the liver. So they just took every hydrophobic molecule they could get their hands on and tried it and just look to see where it would go. First, the backbone, this modification of the backbone now allowed Alnylam to create inclisiran, which knocks down cholesterol enzymes, generating enzymes. And a single dose of this RNAi now lowers a patient's cholesterol for 6 months. So that just shows how long lasting this is, that it can be maintained for so long. So very promising. And then the ionophores can be injected. This is just showing one ionophore across different organ systems and a bunch of different ionophores. Depending on which one you choose, you can either deliver it to the kidney or deliver it to the liver or to both, or any combination kind of in between. Just showing how important that these hydrophobic molecules are to the localization of this drug. But we found one that does well with skin called DCA, as you can see here. And so then Chi actually started to look at all the classic targets that we might want to go after: interferon gamma receptor, JAK1 and 2, STAT1, and found essentially you just have to test a bunch of sequences to see which ones effectively knock down your target, and looking at 25% knockdown or lower is pretty good here. So we can pick the ones that have the best knockdown, and Chi then put them into our mouse model by injecting subcutaneously every week. And he looked at JAK1, JAK2, STAT1, and then a mix of all three. Just put them all together to see if, and then tested that against an interferon gamma antibody. And essentially what we're looking at is the black curve here is normal pigmentation in all the tails. And then the shift left shows, or the black actually is vitiligo, so a loss of pigmentation. And the shift left is how protected that group of mice was from the treatment. You can see interferon gamma shifts it left, as we'd expect, blocking interferon gamma. All of the treatments, all of the siRNAs worked, but the mix of 3 siRNAs against JAK1, JAK2, and STAT1 actually had the best effect. So we're hopeful that if we can now, because RNAi, we can target, very targeted, we can target multiple pieces of the same pathway and maybe get better inhibition without using a drug that has lots of off-target effects and is not specific. This, if you target JAK1, you're targeting only the sequence that only JAK1 has. So there is no, like a JAK inhibitor targets a little bit of all the JAKs, right? And it might prefer one. Here with siRNA, you're only targeting one. But then we can mix them to get a better effect. So then Chi said, I wonder if I can develop a dual targeted siRNA. Can we actually have a single molecule that targets 2 different sequences together? Because that's much simpler to use as a drug without having to mix them. And the FDA would treat this as a single drug rather than 2 drugs, makes things a lot simpler. So he was able to develop the chemistry in order to have 2 siRNAs on the same backbone and delivered at the same time, and show that they still have all the specificity that you would want, but a single target. Here he's looking at Huntington's disease because they're treating Huntington's patients with this through intrathecal injection, just a control versus JAK1. And we can get both Huntington's and JAK1 in the same molecule. So then finally, this was one more idea. Could we actually develop a skin-tethered bispecific antibody to treat vitiligo? So part of the problem is if you use an immunosuppressive drug and you inject that and it goes all over, you immunosuppress the body. And you only need it in the skin. So actually, when I was at Penn, John Stanley studied pemphigus patients, and he was trying to pull out pathogenic antibodies from these patients. And he was really surprised and frustrated that 9 out of 10 of the antibodies he pulled out of pemphigus patients bind desmoglein 3, but they don't cause blisters. They don't do anything. They just bind and they don't cause any problems. So that was hard to understand how these antibodies cause blisters. But then he said, well, what if we take an antibody that binds but doesn't cause a blister, a non-pathogenic antibody, and use that to deliver drugs? And so I said, well, that might be doable, actually, and that might be a good way to concentrate in the skin. And when Chow, a grad student, came to my lab, he's an antibody engineer, and I said, oh, this project might suit you. This has been sitting for 10 years. We never really worked on it, but because I don't know much about antibodies. But Chow had all this knowledge, and he said, oh, that'd be cool. Let me try it. So first of all, we know that if you treat mice with interferon gamma antibody, you can decrease vitiligo. That shuts off disease. We've talked about that many times, right? The problem is there is a human interferon gamma targeted antibody that's been in clinical trials, and it works for HLH, actually. But it immunosuppresses so much that these patients, 56% of them get pretty serious infections. So having interferon gamma systemically suppressed is not safe. And so the idea was, could we develop a bispecific antibody where we use an antibody that binds desmoglein-3, fuse it to an antibody that binds interferon gamma, and then inject it just locally and have it be retained there and not become systemic and immunosuppressed. And this is the concept. So if you just have an interferon gamma binding antibody, of course, it goes into the tissue, but then flows out and gets washed away, becomes systemic, whereas the bispecific would stay there and be tethered. And so Chow actually fused these together. He took PX44, which is a desmoglein-3 binding portion of the antibody, fused them to an interferon gamma binding antibody, or tested the two parentals alone, interferon gamma alone, desmoglein-3 alone. He showed that he could make the bispecific, which is a little bit heavier. And then he showed here on the upper right that the bispecific antibody binds, let's see, what is it doing here? This is competition interferon gamma ELISA, so it binds and neutralizes ELISA just as well as the interferon gamma neutralizing antibody alone. And then here on the bottom, he's showing binding to keratinocytes and shows that the bispecific antibody binds to keratinocytes just as well as the desmoglein-3 antibody alone. So the bispecific's still doing its job after it's been fused together. And then he actually, this was super cool, I've never done anything like this, but he went to the small animal imaging facility, labeled his antibodies with radioactive iodine, and then just injected it into the right foot pad and then followed it over time to see where they went. So on the top is the interferon gamma binding antibody alone, and on the bottom is the bispecific antibody that's tissue tethered. And you can see with the right foot pad injection in the interferon gamma antibody alone leaks out and goes all over the body and really stays systemic kind of for a long term, 48 hours or more. This is similar to what you would do with any of our subcutaneously injected antibodies for psoriasis or dupilumab or whatever, right? You inject it sub-Q and it goes everywhere. Whereas the tethered antibody, actually he injected, and while there was some leakage out 4 hours later, longer term, 1 to 2 days later, it's not in the systemic circulation anymore. So what gets into the systemic circulation actually then goes to skin, and most of it actually stays in the foot pad, so it stays local. So here you can see the bispecific antibody is retained in the foot pad much better than the non-tethered antibody. And you can see the non-tethered antibody becomes systemic and all over the body more than the other. He's got a lot of studies showing this, that the systemic immunosuppression is significantly reduced by the bispecific, that I just don't have time to put in here. Here's a movie just looking, where this antibody is going. On the left is the interferon gamma antibody alone. On the right is the tethered antibodies, again, showing how well it stays just locally where it's injected, and showing that the non-tethered antibody is in the circulation, in the heart and everywhere else. Whereas the tethered actually, what does come out still goes back into skin again afterward. And then he showed in our mouse model that if you just inject one foot pad with the tethered antibody, you can protect just that one foot pad from vitiligo, and it doesn't protect the other one. So on the top is just a desmoglein-3 binding antibody that does nothing for anybody. It doesn't block interferon gamma, doesn't have any effect on vitiligo. Here's an anti-interferon gamma non-tethered antibody, protects both foot pads because you inject it into one, becomes systemic and protects the whole body. Has a very long half-life, so it's like a biologic using the foot pad for the injection site. And then you can see here with the tethered antibody, we see only protection in the treated foot pad and not the untreated. And so looking at the delta between the treated foot pad and the untreated, you can see that the only place where we see protection in one and not the other, indicating that it's not becoming systemic, is in the tethered bispecific antibody. All right, so that's it. This is a summary. Of course, we can target interferon gamma signaling. We can target IL-15 signaling. We've got hundreds of new pathways that we're looking at to try to develop new opportunities that work even better. And then we can target these in a lot of different ways now. We can use conventional antibodies. We talked about IL-15. We can use bispecific antibodies that tether to the tissue specifically and avoid systemic immunosuppression. And we can use next generation siRNA with long-term durability and tissue targeting. And so I have to thank lots of people because I don't pipette anymore. And these are the people that do the work. They're really brilliant, everywhere from undergrads to grad students to MD PhD students to dermatologists from all over the world who come and spend a few years and really enrich our group. We have clinical trialists in this group. We have basic scientists in mice. We have people who are blistering our patients and everybody in between. And lab meeting is just a whole lot of fun where we're all sharing ideas really across the spectrum. Collaborators who are really required to do this work. I'd like to highlight Manuel Garber because finding a computational biologist who will help you is worth his weight in platinum. And Manuel was so excited about the blistering technique that he's converted half his lab now to doing comp bio for skin diseases. It's been a great opportunity. That's it. Yeah. Any questions?
Anyone have questions?
Sure.
I'm wondering if it's known what the antigens are that CD8 T cells are.
Yeah.
Particularly early on.
Yep. Question is, what are the T cells recognizing? What are the antigens recognizing in vitiligo? And this actually goes back to why I decided to study vitiligo in the first place. Vitiligo, we actually had a big leg up because melanoma immunotherapy has been studied for 20+ years. And it turns out that the melanoma antigens that are targeted are known very well because they were actually used for vaccination treatments in patients, and it gave them vitiligo. And it turns out that the vitiligo antigens are the same. So MART1, GP100, tyrosinase-related proteins 1 and 2, those are all the targeted antigens. And we have good immunologic tools to actually be able to find T cells that target those antigens. We see that they're enriched in vitiligo, they enter the skin, and those are the targets in vitiligo. But there's probably just the tip of the iceberg. You know, those 5 antigens and many peptides within those antigens are the targets. But there's still a lot more to know. So if we take and do our blistering and we look at all the T cells in there using tetramers against those antigens, we can use 4 different antigens and we see maybe 1 out of 20 T cell clusters binds to that antigen specifically, which really tells us that a very small population of the cells is binding what we know about, meaning most of them are binding things we still don't know. Probably lots of antigens we don't know about. There's also some old, not that old, I guess, relatively recent data in type 1 diabetes. So for a long time, we thought that autoreactive cells in diabetes were attacking insulin because you can take cells out of the pancreas, grow them up, give them insulin, and they make gamma and they get angry. But it turns out that there are some new studies showing that if you take insulin and fuse it to chromogranin, now that makes them turn on 10 to 100 times more. And that's called a neoantigen, where you've taken 2, normal antigens, you stick them together and you make something new that the immune system has never seen before. And that could be what's happening in vitiligo as well. We know stressed melanocytes induce this whole process, and it may be through neoantigens. So yes, we know some, and there's probably way more that we don't know yet. And then the hope is we can flip this on its head and develop melanoma immunotherapy. If we're very good at killing melanocytes, so why not use that for good?
The neoantigen idea is interesting because melanocytes are highly mutated. There, but then we can spread it.
Yeah, so the point, just for the listeners, is neoantigens are interesting because melanocytes get wonky. They get mutated by the sun, and antigens could be changed that way, and then it can spread to other things. Even better, I'll show some slides tonight that vitiligo can be induced by chemicals. There's chemically induced vitiligo. We've known it for almost 100 years now, and we know a lot of the chemicals, and monobenzone is one of them. That's been turned into an FDA-approved cream. And the way it does it is it induces stress within the melanocyte. And the melanocyte undergoes autophagy and burns a lot of energy and reactive oxygen species. And it's that autophagic process that likely fuses those proteins together and creates neoantigens. And so we think actually even better, you can induce neoantigens by using these chemicals that modify the melanocytes even more and make the disease worse. It's a lot to do. Yeah.
It was a great talk. Thank you. I was wondering if you thought about using your bispecific antibody technology to use with the IL-15 or IL-15 receptor antibodies to target the resident memory T cells, specifically in the plaques of vitiligo. And if you think that might give a more durable response than just targeting interferon gamma in those plaques.
Yeah. So the question, yeah, so everybody heard that because it was in the microphone. So one of the things that we were a little surprised by when we used the bispecific antibody is how short the half-life is. So we thought that we'd give one injection and it would last for 30 days because of the turnover and everything. And it actually turns over very quickly. One of the benefits of that is that means that your antibody and whatever it's attached to gets internalized and degraded by the keratinocytes. So it's like chewing everything up. So we think what we're doing against the interferon gamma is binding it, pushing it into a keratinocyte and getting it chewed up, and then keeping doing that. So you degrade this driver. And so IL-15, this is in revision now. So we added that to the discussion that IL-15 might be a great target for the bispecific approach too, because IL-15 is made by the keratinocytes and so is desmoglein-3. So we can just cause sequestration of that IL-15 within the keratinocyte degradation for the same reason you just said. The other thing that we thought about a little bit more is what if, what we kind of set out, what we wanted to do is maybe be able to inject it IV and have it just home to the skin, all the skin, and stay there for a long time. And to do that, maybe we should target something in the basement membrane that doesn't turn over, as much collagen, some of the collagens 4, 7 or something, as long as we don't disrupt them.
Two of you, don't you? So you didn't talk about sort of anti-PD-1, chemo side effect, vitiligo, number one. And number two, I want to push you a little bit on the kind of the localization. One of the hallmarks of this disease, alopecia areata, as well as this regional localization. But as we see with the chemo responses, you still get this patchy response. When you first infuse the CD8 cells in your mouse model, you get a patchy response. Mm-hmm. Do you believe that there's a sort of a selective absence of TRM in some places? There must be more to the fixed localization than just the resident memory. So
that's a big question we want to answer. And we have a great opportunity to do it, right? We've got a great model to try to understand why you get these fixed lesions that are not spreading more. I mentioned that if you deplete T-regs in the mouse model, you don't get the fixed lesions anymore. They just all turn white. So we think T-regs could play a role in controlling that inflammation. But the simplest explanation is that you just get patchy infiltration, and that's where the lesions come from, and you have nothing in normal skin. But that's not what our data showed. So if you look at the single cell data, we looked at non-lesional versus lesion, and the non-lesional skin has a very strong inflammatory signature. It's just subclinical. So you have way fewer T cells in there, but the T cells that are in there are making interferon gamma at a much higher level than you see in healthy skin. And what's really cool, I didn't get into it, but it's in the paper. You can look at, because it's single cell RNA sequencing, you can look at who's making gamma, and then you can look at all the gamma responsive genes to look at who's responding to gamma. And what's cool is in lesional skin, almost everybody's responding to gamma, including the melanocytes, telling you that the T cells are making gamma close to a melanocyte, so the melanocyte's getting signaled. In non-lesional skin, the T cells are making gamma, the melanocytes are quiet, suggesting that the T cells are not near the melanocytes in non-lesional skin. They're still making gamma, but the T-regs are sensing it. So it's like the T-regs they know that the effectors are making gamma that they want to kill, and they're regulating them and dampening them so they can't get up to where they need to be to cause the problem. Keratinocytes also. So lesional skin, keratinocytes, melanocytes all responding to gamma. In non-lesional skin, they're not, and only the T-regs are responding. So it suggests that the T-regs are playing a really important role. And everybody, when I give this talk, I have to be reminded, because people are like, what do you mean you're talking about T-regs working in lesions? Like the lesions are there because the T-regs aren't working. But the reason they're lesions and focal is because the T-regs are doing a great job. They're just not enough to keep it, completely silent. So I think that in a short answer to your question, I think that the CD8s are everywhere. I think they're all over your skin, and they're trying really hard. And I suspect even in even in patients who don't have vitiligo, but are genetically predisposed, probably also have that inflammatory, subclinical inflammatory signature there. It's just that the Tregs are preventing it from happening in those areas. It's a really important question. And what's also interesting is there's really good genetic data now for vitiligo. We know 60 GWAS risk alleles. And what's weird is that those with a high BSA, really severe vitiligo versus those with very focal, small amount of vitiligo, genetically they're identical. The genetics don't, it's not the more risk alleles you have, the worse it's going to be. It really just indicates who's going to get it. And then once you get it, something after genetics works. Right. So PD-1, a great opportunity to study, in humans, how vitiligo occurs, right? So you have melanoma, you block PD-1. I think of it as like cutting the brakes on the car, and then everything goes out of control. And even then it's patchy, but it's active. I'll talk about active versus stable vitiligo tonight. And confetti vitiligo is a sign of activity. You just get 1,000 1 millimeter dots that then fuse, but you can see that it's just going nuts. That almost always happens with PD-1 inhibition. So it tells you that the fact that PD-1 inhibition is patchy tells you that it's not PD-1 preventing the spread, right? I still think it's Tregs. And again, the Tregs chase them in and they just get overwhelmed, I think, in the lesions and can't fully control.
Okay, thank you so much.
Yeah, thank you.
I think for that. Oh, sure. I don't know if we have time for the
Okay.
chat, unfortunately,
Okay.
because I wanted to give people time to talk with you
Sure.
downstairs.
Yeah, yeah.
Okay.
Sure. Thanks.
Is there something in the chat?
So,
Can't find it.
I want to invite everyone. Everyone's invited. You want to have questions for Dr. Harris?
I told you I could talk for hours.
Right.
You're gonna regret the same.
Yeah.
That's exactly what we're gonna do.
All right, okay.
Very good.
I'm
I
going
just
to,
need to grab that little.
yep, I'm gonna